Hello everyone, my name is Khadija Yunus and I did my graduation in Doctor of Veterinary Medicines. Then I did my master in animal reproductions. Now I'm doing PhD in integrative bioscience, and today topic of my presentation is investigating the impact of zearalenone on reproductive performance of dairy cattle. Here's the introduction. The livestock sector, in which the agriculture contribute about 5.5% to the United States GDP, and in the agriculture sector, the cattle population is 87 to 89 million a head per year, and milk production from the cattle is 226 billion pounds annually, and meat productions 12 to 13 million metric tons annually in the United States. Here are some properties of the zearalenone. So zearalenone is a mycotoxin that is produced from the Fusarium fungi, and zearalenone have a estrogenic property, like it look like a estrogen, like they have the same structure, so it has a estrogenic property. The molecular formula of zearalenone is C18H22O5, and zearalenone is a heat stable, like it can be stable up to 150 to 160 degrees centigrade, and zearalenone is organic, so it soluble in organic solvent like ethanol, alcohol, etc. So there are four types of zearalenone: alpha zearalenone, beta zearalenone, alpha zearalenone, beta zearalenone. This is a schematic representation of the effects of estradiol and zearalenone on the reproductive system. On the left side of the picture, there that is a normal picture in which estrogen receptors are present on the uterus, and estrogen comes and attached to the estrogen receptor, and the main function of estrogen is to produce mucus in the female tract, and also myometrial contraction in the female tract. So this is a schematic diagram where estrogen performs its normal function. On the right side of this diagram, when there is no estrogen present, or there is a minimum production of estrogen in females, like in animals or in humans, the zearalenone that comes from the feed at, attached to the estrogen receptors, because zearalenone and estrogen have the same structure, so it estrogen receptor attached accept the zearalenone, and when zearalenone attached to the estrogen receptor, they perform many functions, like they increase the abortion rate if the cattle is pregnant. They cause the cystic ovaries, like there a lot of cysts that can't ovulate present on the ovaries, and decrease the fertility rate. Also delayed ovulation, because when there is a low amount of estrogen, there is no LH surge, luteinizing surge, that cause the ovulation, so it cause delayed ovulation and forms the many cysts on the ovaries. And if there is no ovulation, then this decrease the conception rate, and then it's develop also induce the developmental abnormalities in the calf. Here are the impact of zearalenone on the cattle, like it causes estrus disturbance, reduce fertility and embryonic loss. Then it causes hyperestrogenic in cows, and it decreases their milk production in cow, and hormones fluctuation mainly are estrogen and progesterone. It also impact on the uterus, because it produce mucus secretion and cause myometrial contraction in the pregnancy, so it's mainly effect on the uterus. Also estrogen receptor present on other part of the body, but there is main effect on the uterus. They also change the morphology of the ovaries and uterine mucosa. Here are three objectives of my research that I performed in my master's degree. First, zearalenone prevalence in the feed, hormonal assay in the blood sample, and the impact of zearalenone on reproductive performance. So first, I collected the feed sample, checked the prevalence of the zearalenone in the feed. Then I collect the blood sample from the cattle and find out the normal hormonal assay and their changes after taking the zearalenone, and the impact and the direct impact of zearalenone on the reproductive performance. Here's the material methods. So on the experimental station, the study is planned to be conducting on two farms. On farm 1, that is semi-intensive farm, that is livestock experimental station, University of Agriculture Faisalabad in Pakistan, and the farm 2, that is intensive cattle farm, that is Colonel Osama Dairy Farm Faisalabad in Pakistan. So say in semi-intensive farm, there is no toxin binder they are using, so they just use the feed and fodder, just giving to the cattle. And in farm 2, they are going to use toxin binder mix in the feed and then give it to the cattles. Here are the groups that I form. There are four groups that I selected, and I give the name group A, B, C, and D. And in group A and C, I took the sample from farm 1, and number of parities in group A and C is 3 and 4. Then in group B and D, the number of parities is third and fourth, but I took the sample in farm 2, that is intensive farm, in which toxin binder are used in the feed. I took the two types of feed sample will that is collected from the each farm. The one is the fodder and the other is concentrated feed. Zearalenone was detected through the commercially available ELISA kit. So here's the picture of the feed sample that I collected from both farms. One is fodder and other is concentrated feed. Here's the procedure that I made the feed sample to prepare for the ELISA. Firstly, I collect the feed sample, then mix the well ground samples until it becomes passing a 20 mass sieve, then suspended 5 gram of a sample into 25 ml of 60% methanol water solution. Then I vortex extraction for 5 minutes. After that, I centrifuge at 4000 R per minute for 5 minutes. Then I filtered the extractions and then transferred the supernatant 40 microliter to a new centrifuge tube. After that, I add 960 microliter of the distilled water and then perform ELISA on diluted sample solution. So here's our sample is prepared for the ELISA. Here's the picture of performing ELISA on a feed sample. So I already prepared the feed sample for the ELISA. So I took the sample solution plus the standard solution and pour onto the ELISA kit. Then I add enzyme label conjugates on it. Then I add antibodies. After that, I incubate at ambient temperature, and then I seal the plate with the cover. And after that, I discard contents. Wash 3 times with wash buffer solution that is already given by with the kit. After that, I add the substrate solution and incubate at ambient temperature, seal the plate with cover, and then I add the stop solution. After that, I measure at 450 nanometer of each well. Then I add the ELISA kit in the ELISA reader and measure the recording. Here's the blood sample that I took from the cattle. So blood samples were collected from the 40 animals in Equatorial, 20 animals from each farm. After extract the serum and stored at minus 20 degrees centigrade for further analysis. Before that, and here's the reproductive parameters that for reproductive parameters that I find out from this experiment. Here, first is fertility record of the cattle, then ovarian cyst record, then low conception rate and delayed ovulation due to the effect of the zearalenone, then abortion rate in the cattle, reproductive abnormalities, and then early abortion rate. After that, there is a hormonal assay analysis. First is estrogen because the main hormones that is affected by the zearalenone is estrogen, and the second one is progesterone that is also fluctuated due to the zearalenone. And I find out these two hormones levels will be detected in the serum samples using commercially available ELISA kit. Here's the picture in which I detect the zearalenone concentration. So uh, the normal range of zearalenone is 300 ppb. If zearalenone concentration more than 300 ppb, there's negative effect on the cattle. If there is a less concentration of zearalenone in the feed, about 300 ppb, then there is a less effect in the cattle. So if we have a zearalenone concentration more than 300 ppb, then we have decreased reproductive performance, increase the abortion rate, increase the ovarian cyst, and also with that increase the developmental abnormalities in the calf, also decrease the conception rate and increase the delayed ovulation. So zearalenone concentration in which less than 300 parts per billion, uh, uh, there's a different effects in which we have like a little bit increased reproductive performance compared to the other one, and less abortion rate, and less ovarian cyst, and less ovarian developmental abnormalities in the calf if the cattle is pregnant, and increase conception rate and decrease delayed ovulation. Here's the graph diagram in which one detect the zearalenone less than 300 uh, 300 parts per billion and zearalenone more than 300 parts per billion, and their effects on the cattle. So final conclusion, zearalenone contamination that is more than 300 parts per billion has significant negative effects, that is semi-intensive systems are at high risk because they are not using any toxin binders in their feed that they give to the cattle, and so they have the negative effect on the cattle reproductive system. Besides this, there is a strong link between feed contamination, hormonal imbalance, and reproductive failures because feed have the zearalenone, and that is a mycotoxins. If the cattle eat that feed, they have the hormonal imbalance. If the hormones are going to be imbalanced, they have the reproductive failures in the cattle or in other animals. Here are some future directions. that is regular monitoring of zearalenone levels in the cattle feed, implement strict feed quality control and storage practice, and also use mycotoxin binders or detoxifying agent in the feed. Also develop rapid detection method, field based kits for early diagnosis, investigate long-term effect of zearalenone on fertility and herd productivity, study molecular mechanism of zearalenone interaction with estrogen receptors. So these are the future directions that we have to going to develop if we are going to stop the zearalenone effect on the cattle or other animals, even in the human beings. We have to use some toxin binders. We have to check the zearalenone level in the cattle feed. We strictly check the quality control and storage practice, and effect of zearalenone on fertility and herd productivity, and we have to use zearalenone interaction with the estrogen receptors, and also we I want to add one more, that we also have to use other detection method that are more easy to find, and like ELISA is a little bit expensive for the farmers, so we have to find out other zearalenone detection methods, so everyone can do that and find out is the zearalenone present in their feed or not, or is they have to use any toxin binder in their feed or not. So here are the reference that from that I took the informations, and their research guidance. These are also the references that I took the information from these different, uh, and thank you so much.